Hi Nat5s, so we are still on variation and inheritance, and in particular we're going to start looking at the genetic terms that are used when we're talking about these. You need to have these right, otherwise you're not going to understand the questions, because quite often they use these terms specifically, and also it's just content that you need to know. Right, so we'll start with the word gene, because that's going to be rather important for genetics. So this is a section of your chromosome that codes for a particular characteristic. We've already looked at that term and we've mentioned it when we've talked about chromosomes anyway, so it's essential that we have this in your head. Now this is actually obviously a very nice DNA spiral. It's a slightly dodgy diagram of a gene, to be honest, because we have across here, these are your base pairs and you should recognise those, and remember A to T, G to C, the straight lines and the curly ones. That's a rather not straight line A, but you know what I mean. And most genes though are going to be thousands and thousands. We don't even talk about genes normally in terms of base pairs, we normally talk about them in kilobase pairs. Okay, so we're talking thousands of base pairs. But the basics is always the same. So we've got our cell, and inside the cell we've got the nucleus, and inside the nucleus we have the chromosome. The chromosome is a huge long string of DNA, and sections in that are the gene. The next new term, and a really important one, is the term allele. So what we mean by that is that each gene can have several different forms that will code for different versions of whatever it's coding for, and obviously each gene codes for a protein because that's what genes do. So here's an example. We've got a gene on this chromosome which is an allele for purple flowers, and on another copy of the chromosome, so the same kind of stretch of stuff, we have an allele for a different colour of flowers. If we put that into something that you might recognise, let's take for example the blood groups on you. Okay, so our blood groups have an A allele, a B allele or an O. We have three different possible alleles on the chromosome that we have that has blood typing. Okay, so you could get an A from mum and an A from dad, or you could get an A from mum and a B from dad, or you could get, say, an A from mum and an O from dad. You can see how this starts all to get quite kind of complicated. Now it doesn't actually matter, so say you got an A from dad and a B from mum, that would still be an AB. Okay, so you could also have a B and an O, or you could have O and O. And these would give you different blood groups, but you would still fit into distinct categories. So this is monogenic again, so this is a single gene. Okay. When we're trying to talk about crosses, because that's what we're going to be looking at properly, then we put them into generations, and the generations that you need to know are P is for parental, F1 is for the first filial, F2 is for the second filial. Okay, it comes from Latin for sibling. Okay, so we've got, in this one, here's our parents, so generation P, and you've got a tall plant and a short plant. And then our F1 generation, so our first generation of siblings, of all of the seeds basically, and they were all tall. And then we've got an F2 where they've taken two plants from the F1, crossed them together, and they've got a whole pile of siblings as well. Okay, so what we got from here. So when we try to actually talk about what's going on, we're going to use the terms phenotype and genotype. Okay, phenotype is in some ways the easiest one to deal with because it's what you see. Okay, so it's going to be the appearance of the characteristic. Now what you see could be quite complicated, like your blood group. You can't actually see what blood group you have, but if you do a test you can find out if it's an A or a B, and so that's still your phenotype. Okay, so in this case our characteristic would be petal colour, and our phenotypes are all of the different colours that we have. Okay. To make that understandable from a sciencey point of view, we need to make sure that we've made it clear exactly how many we have of each type. So we put them into ratios. Now this is where you're going to be asked to cancel down to the smallest whole number ratio. Okay, so if you're not happy with that, make sure that you've gone back over how to do ratios. So in this case, our phenotypic ratio, let's look at this one first because it's kind of the most straightforward. We have got tall plants and small plants. Don't worry about these yet, we're going to look at these in a minute. And they gave all offspring here, and they were all tall. so our phenotypic ratio is just all tall. Okay, but if we look at this one over here, and this time we're looking at the colour of peas inside pea plants, so we've got a yellow one and a green one. Okay, now the yellow one and the green one, when you cross together, they all came out yellow. So this would just be fine. You would have just said all yellow. But then when you cross two of these yellow ones together, you got something slightly different. And that's where the phenotypic ratio comes in. You would say here, 3 yellow to 1 green. Okay, and you're going to have to be able to work out how many of these are likely to happen, but that's a little bit further on. We also have our genotype. Okay, so the phenotype is what you can see. The genotype is what is actually inside you. So what you're carrying on your chromosomes, the alleles that you have present. So in this case, these are the letters I was saying. So in this fish here, they have a big A and a little a. And this fish also has a big A, little a. So if I looked at their chromosomes, inside their chromosomes, assuming these were 2 copies of homologous, that means the same chromosome, this one would have had a big A on that one and a little a on that one, and the same would be true on this one. Okay, so this one, which we know is female because they're producing eggs, they only pass on half of their DNA. You know that. You know that you only have haploid cells when you're producing sex cells. So you either get a big A going to an egg or a little a. You've got a kind of 50-50 chance of what you're going to get from this. And the sperm being produced by this male are going to be the same. You could either have a big A or a little a. Now that means your potential crosses are, I either get big A, big A, little a little a, big A, which to be honest I'm just going to write again as big A, little a, because that's a convention and it's easier because then I know it's the same, and little a, little a. Okay, so this is the genotype. These ones here are the genotype of each of those fish. So you'll notice that we had 2 different forms of the letter there, a big letter and a little letter. Now the reason for that is because we know that some alleles will show up even if there's just one of them there. Okay, and that's called a dominant allele. So to show that it's dominant, we give it a big letter. Okay, and the only thing you've got to do there is just be careful that you've picked a letter that makes sense. So like I've said here, big A, little a is good, W is not so good. So A is fine, B is good, C is rubbish, because you can't be absolutely certain in an exam that your C's are not going to end up looking like the same. Okay, so go with things which make sense to use and don't use ones that don't. Oh, that's gone over the top. Never mind, you can still see it. So our second type is recessive. And what we mean by that is that this allele, this form of the gene, needs 2 copies in the genotype to show up in the phenotype. So reminder, we would use the right kind of letter and not the dodgy ones. Okay, and this was from our slide a bit earlier. We had all yellows, but the yellows threw up a green. Now that shows you that this one must be recessive. Okay, and that means that it must have 2 copies of this gene which was hidden in this yellow one. So actually you could even figure out these ones must have had a little a in there, and this one must have had a big A as well, because otherwise they didn't show up as yellow. So all of these ones have a yellow, big yellow, but you actually don't know if it's a big A or a little a in there. You can't tell unless you do something called a test cross, which we're going to talk about later. So to describe these with more words, sorry, but there's quite a few. If you've got 2 copies of the same allele, then we use the word homozygous. Okay, it's a pretty common root word. So homo means the same. Zygous actually comes from the zygote, which is the fertilized egg. So what we're saying is that inside the fertilized egg you had the same copies of the gene. So it's either going to be homozygous dominant, big A, or homozygous recessive, little a, or whatever other letter you're using. Okay, so if you've got homozygous, it should not surprise you that we have heterozygous. So this time we have 2 copies of the allele, but they are different. So big A, little a. Last term, This is the genotypic ratio. So if we've got a phenotypic ratio, that tells us all of the things we can see. So here's another example, very similar to the one we had before, a 3:1 phenotype, 3 purples and 1 white flower. But the genotypic ratio, if you can actually see inside the alleles what you have, is a little bit more complicated, because these 3 purple ones that we have here are not actually all the same. What you have is 1 that is big P, and then you've got 2 which are big P, little p. So even though our phenotype is 3 for purple, our genotype is a 1-2 here, and then we've got our final 1 down here, which is the homozygous recessive. You'll notice this is P's, but which is not one I would recommend to use as a letter, but if you're typing it's generally okay. So this down here is our phenotypic ratio, and that's your terms that you need to have sorted.